You are listening to the PFAS Research and Remediation podcast series, produced and created by Arcadis, with funding from the Environmental Security Technology Certification Program, ESTCP, grant number ER23-7692, through the United States Department of Defense. All opinions, interpretations, and conclusions expressed belong to the hosts and guests and do not represent views or policies of the Department of Defense, Arcadis, or guest affiliations. In this first season, we're focused on PFAS and interview a broad panel of experts who have each contributed to the growing knowledge base around remediating this emerging chemical of concern.
Today, we'll be speaking with Dr. Jason Conder, a PhD environmental toxicologist and chemist, a principal scientist that has been working with PFAS for over 15 years. He is a principal investigator on multiple SERDP and ESTCP projects, but we will focus on his project, Critical Data for Assessing the Marine Toxicity and Bioaccumulation of PFAS. I'm Dr. Theresa Guillette, a PFAS technical expert with Arcadis, and I'll be your host today. So, environmental toxicologist and chemist. In grad school, I was told you had to choose one. I really loved analytical chemistry and I really loved toxicology, but my committee was very much focused on go towards one thing and just be good at it. And now, sitting across from like a successful scientist that does both, how has that really impacted your career?
That's a great question. You know, I loved chemistry growing up. I loved ecology. I was really into biology. Sort of the intersection of those, you know, in environmental sciences, environmental toxicology and chemistry. So I took a lot of chemistry in school, as you know, almost got a minor in it. I was really excited about it, but using it as kind of understanding ecology and toxicological problems, that was what really kind of drew me to my career. And I just kind of made my own way in my graduate work. I knew what I wanted to do. I knew, you know, it involved a lot of serious analytical chemistry and environmental chemistry to understand the sort of questions and research ideas that I had, but it also involved a lot of hardcore ecology and biology. Again, I just sort of did my own thing and it worked out really well, and I get to sort of dabble in a couple of really exciting fields. And my practice over the years has just revolved around the whole how clean is clean question. That's really been at the heart of what I do for clients and my research work, understanding how chemistry affects living organisms and how, at the end of the day, what do we do about it at contaminated sites or working with particular products or processes.
That's really interesting, hearing a little bit about your pathway into science. Specifically, how did you get involved in the SERDP and ESTCP programs?
That goes back to sort of early career helping out other scientists at a consulting firm called Environ for a few years. And they had some ongoing work with SERDP and ESTCP projects. And it just started out as a young scientist, as a consultant, helping out here and there. And eventually I got in positions to where I took more of a leadership role. And eventually I started coming up with my own proposals for SERDP and ESTCP and trying to win those. And over the years, working with many collaborators on different projects, you kind of getting in front of the SERDP ESTCP crowd, you know, winning my own work and my own projects and directing my own research that worked into the programs.
Now, before we dive deep into your marine project, can we talk a little bit about the basics of PFAS bioaccumulation in the environment?
Sure.
Well, just generally, how do PFAS bioaccumulate in the environment?
PFAS, like any organic chemical, will accumulate to a certain degree in a living organism. So it will build up in tissues, basically through diffusion. And when we first started coming at this in the early 2000s, I was involved in some work with DuPont at the time, working as a consultant. There was a big investigation on how are these things accumulating in polar bears in the Arctic. And that was sort of my piece in the work, is to understand how it was moving through a food web. And one of the key things that was really apparent in the early work back in the early 2000s, these chemicals weren't behaving like the traditional hydrophobic organic compounds that, as an ecotoxicologist, we deal with day in and day out. The things like PAHs, PCBs, organochlorine pesticides, those chemicals like fat, they like lipid. They accumulate, they'll diffuse into an organism and accumulate in the fats. PFAS was different. It wasn't accumulating in the fats. It tends to behave a little bit more like a fatty acid or a cholesterol, something like that. It tends to stick to protein. It's a proteinophilic molecule, at least the ones that we know a lot about, like the perfluorocarboxylic acids, the perfluoroalkyl sulfonates, PFOA, PFOS, those sorts of chemicals. They like proteins for whatever reason. They don't like fat, so they'll build up in muscle, blood, liver, kidney, things like that. And that was really what threw everyone for a loop, because when they came along, we looked at them and said, these chemicals aren't very hydrophobic. They're not going to behave like the traditional chemicals that we track. They're probably not going to be a big deal because they're so water soluble. They don't like fats. This is not going to be, an issue. But lo and behold, these chemicals were just different in the way they bioaccumulated. They like those proteins. And a lot of them, particularly PFOS and the longer chain compounds, are pretty bioaccumulative to the degree that we are concerned about how they build up in food webs, build up in fish and plants and other animals and into the food chain. So I tell people there's no one-size-fits-all approach for understanding bioaccumulation for these groups of compounds. You have the short ones, which can build up in plants. You have the longer ones, which can build up more in the animals. And then you have a gradation in between, where the middle range PFAS, maybe they accumulate a little bit in the plants, maybe they accumulate more in fish and other mammals. So again, there's no simple rule like what we have for, say, PCBs or petroleum compounds, which we know tend to build up a little bit higher in the food web in many cases.
What are the differences in bioaccumulation for those short chain versus long chain compounds?
The short chain compounds, when those are taken in, they're basically eliminated more quickly than the longer chain. The short chain won't bind as tightly to proteins and things like that in a typical animal body. But what's interesting is, in a plant, for example, the short chain compounds tend to be more water soluble. So they're taken up by the roots of the plant and will tend to bioaccumulate in the upper portions of the plant. Think like leaves and stems and things like that, more than the longer chain. So with the shorter chain, we tend to worry about accumulation in plants a little bit more, whereas the longer chains, we tend to worry about more in terms of animal bioaccumulation. They don't accumulate in plants as much.
Why do we need this knowledge about bioaccumulation to be important for our understanding of PFAS toxicology in wildlife?
We need to understand bioaccumulation of these compounds because that gets back to the how clean is clean question. So right now we have hundreds and hundreds of sites across the U.S., thousands in the world, where PFAS is present in soil, water, sediment, groundwater. We need to understand how to clean that up, which sites to prioritize in terms of cleanup. And when we are at a site, where do we stop in terms of cleanup? Our analytical chemists are extremely great at detecting very small levels of these compounds in the environment. But just because we detect something doesn't mean we need to clean it up. Doesn't mean it's a risk. Paracelsus, the father of toxicology, tells us that the dose makes the poison. So it's not the detectability according to an analytical chemist, it's presence and the dose to an organism, a person interacting with the site. So, and to do that, we basically need to do what's called chemical risk assessment. So that involves understanding quantitatively how things bioaccumulate from sediment, soil, water, what have you, into plants, into animals, so that we can understand the levels that we need to clean up to in the abiotic media. How clean does the soil need to be? What concentration is acceptable so that it won't bioaccumulate in plants, animals, things like that, at levels that are unsafe for consumption by higher trophic level wildlife or by humans that may be interacting with the site, consuming fish, consuming plants, things like that.
And what are the main drivers for needing a risk assessment at a particular site?
Yeah, usually for ecological, the drivers are presence of PFAS exceedance of usually a screening level, if there are any, and then the presence of the potential receptors. Things like, do we have wildlife there? Do we have plants? Do we have enough habitat there that it is of concern to stakeholders?
Now, in several of your presentations, I've heard you mention this idea of site-specific ecological risk assessment. What does that mean, and why can't we just apply a broad risk assessment to all sites?
Great question. I'll start by talking a little bit about the human health risk assessment process. In risk assessment, humans are fairly straightforward. We understand how they interact with the contaminated site. We know how much water they drink per day. We know that they're going to spend 8 hours a day working at a site, for example. We have a pretty good idea on how they interact with the site and how we can, say, derive site-specific cleanup approaches to protect humans from potential contamination that may be present. In contrast, ecological risk assessments are all unique snowflakes. Every site is different. You have different types of habitat. You have different environmental features. You have different release mechanisms for chemicals that may be present and transport pathways. You have different species that the stakeholders may be concerned with. So ecological risk assessment is just inherently more complex. We have more species involved. You also have extremely different types of exposures involved. Again, humans, we know they drink water. They may incidentally get exposed to soil at sites by accidentally ingesting dust, things like that. But with eco, you have fish, you have birds, you have mammals, you have plants, earthworms, insects, a wide variety of potential receptors that could be important in terms of understanding risks to the ecosystem as a whole. So when you come at that, you have to say, okay, you need to do a site-specific assessment. So you have to come up with a certain limited list of receptors to think about. Usually you select a few bird species and a few mammal species, for example. You look at plants. Once you have that, you say, okay, what about the habitat and releases of the chemicals at a site could cause potential exposures to those receptors? So all that is very site-specific. Again, every site is different. Every site for eco is its own unique snowflake of a investigation, and that's why the how clean is clean question always differs from site to site when it comes to eco. You can generate generic screening levels for ecological risk assessment, but the first thing we usually do is say, well, let's actually look at what's at a site. What animals, plants, and exposure pathways do you have?
Why have you decided in this project to focus on marine toxicity specifically?
There's a lot of great data out there for freshwater organisms. They're a little bit easier to work with in the laboratory, and usually what folks do is basically controlled dosing studies where they'll add certain PFAS to water, observe what happens with standard test organisms, and based on that data, that's how we set levels that are safe or potentially of concern in water for use in water quality criteria, the screening levels that I mentioned earlier, basically trying to give us a guide for when we have water samples, we measure PFAS, we find it, we know, okay, what do we need to do next? Is this of concern? This might exceed and cause toxicity to aquatic organisms or not? So a lot of that work's been done for a lot of freshwater species. There's not a lot of data out there for saltwater species. That's because saltwater species are a little bit more difficult to work with. You have to have seawater nearby or create artificial seawater. That's not a straightforward process. Marine organisms are just a little bit more difficult to work with in a lab setting, just in general. But the thing is, you absolutely need those saltwater data because the existing information that we're getting from a lot of the preliminary work is that saltwater organisms may not be as sensitive as freshwater organisms. And you have many of these DoD sites, you have many private sector sites that are coastal or may be near the coast or in ocean situations. Think like marine vessels that may be discharging PFAS, where you need to understand the toxicity to marine aquatic life, not just freshwater. So we need to know what numbers in water are safe or not safe for marine life, just like what we do for freshwater.
And it does look like you are doing both single and then PFAS mixture testing. Why do you have a need to do single exposure when aqueous film forming sites have multiple PFAS?
The aquatic life criteria in the US, it all revolves around single chemistry sorts of approaches. You want to know what the criteria might be for particular single compounds. That being said, you never find just one PFAS at a site. They're always present in a mixture. So what scientists do, what toxicologists do to understand how to work with a mixture and the risks of a mixture is a very regimented series of single chemistry tests. And then based on that information, we use a model, usually a toxic unit approach, or sometimes a binary model approach that basically allows us to design a mixture experiment where we evaluate the mixture interactions of the chemicals. But before we can get to that critical mixture test where we can kind of say, are chemicals additive, synergistic, antagonistic? How do we numerically work with those data? We have to have that single chemistry data to be able to design that. So kind of two reasons why we need the data on single chemicals first. One, that's the way most regulatory agencies like to see the data, but two, you have to have that base data to design your mixture study that really tells you how to deal with multiple chemicals at the same time.
Coming back to your how clean is clean question that has been really central to your whole career, how do we anticipate that PFAS are going to change that, given that we know that it's in the rainwater? We know it's at high background levels. I mean, I can go and catch a crocodile from South Africa in the most remote part of the world and still find PFAS in its blood. Like, how do we deal with that?
The analytical chemists, again, they're amazing. They can detect extremely low levels, and they find that this stuff is everywhere. It's hard to take a tissue sample, a soil sample, a water sample, really anywhere on planet Earth and not find at least a little bit of PFAS. They have long-range transport potential. They're persistent. They can accumulate. Again, the analytical chemists are extremely sensitive. And I think going forward, folks with responsibility for PFAS cleanups absolutely have to consider the ambient conditions in their local and regional area. You cannot clean up to non-detect. There is no zero PFAS exposure. It's just impossible to do that. Again, the dose makes the poison. We don't necessarily have to get to non-detect. Some amount of PFAS exposure will not be associated with an adverse effect. Again, that's just a tenet of toxicology. But I think entities that are involved with PFAS site cleanups are going to have to evaluate local ambient background to understand what is really doable at their site in terms of cleanup levels and risks. And in many cases, there may be a few sites where the ambient conditions that are beyond the control of any one party or site owner, for example, are just going to be as they are, and they may indicate low levels of risk. And that's generally, as a society, I think we just have to be prepared for that and manage what we can manage. And in many cases, the ambient conditions are just going to be beyond our control.
Beyond this project, what are the main challenges for ecological risk assessment with regard to PFAS?
The biggest thing that I see coming in the next few years, maybe decades, is really how to handle PFAS that we know might be present that we can't yet routinely measure with commercial analytical techniques. The academic analytical labs are telling us there's hundreds to thousands more PFAS at many of these PFAS sites. We know that AFFF has a lot more chemicals present besides just PFOA, PFOS, and their carboxylic acid and sulfonic acid brothers and sisters. What do we do in terms of ecological and even human health risk assessment? As a risk assessor, I need to know potential adverse effects and which doses are associated with those. I need to understand bioaccumulation rates and solubilities and where we find them in the environment. We can't understand all those numbers for all those chemicals. So I think the big challenge for us right now is to understand how to evaluate the risk of that material, those substances, again, that may be present. What's the risk? Or develop some sort of paradigm similar to what we might have for petroleum hydrocarbons or other chemicals, where we have a subset of particular compounds that we evaluate the risk. And if we address those, we can rest assured that for the other compounds that might be present, they'll be assessed, they'll be taken care of too, if we focus on PFOS or PFOA, for example.
Well, great. Thank you so much, Jason.
Yeah, happy to. Yeah, fun stuff.
Thank you so much for listening. This podcast was funded by ESTCP and recorded by Arcadis. The interview was conducted by Dr. Theresa Guillette, and our guest today was Dr. Jason Conder. If you're interested in more information on Jason's project, please see the ESTCP website for project number ER22-3214. If you conducted your own research on PFAS and are interested in sharing your work, please email Theresa Guillette at teresa.guillette@arcadis.com. That's THERESA.GUILLETTE@ARCADIS.com. And please keep an eye out for more episodes coming soon.